My name is Jeff Rossibara. I'm fortunate enough to be the faculty advisor for this great group of graduate students. Fortunately, I didn't actually have to do any of the work organizing any of this, and they've done a great job, as several of the speakers have noted. It's my pleasure to introduce our speaker for the last talk of the afternoon session, Sarah Hearne. Sarah did her PhD at the University of Sheffield, followed by a postdoc at CIMMYT, and then time as a scientist in IITA, where she worked on a number of different crops in Africa. She's now back at CIMMYT as a principal scientist, leading, among other things, the Seeds for Diversity program that she'll be talking about today.
Perfect, thank you. Am I on? Yeah. Okay, so thank you very much to the organizers for inviting us to talk to you about this project. I'll try to keep to time. I realize we're all somewhat caffeine deprived, me included, so let's see if we can race to coffee. Okay, so I want to talk to you about between gene banks and breeding programs. Within CIMMYT, so the International Maize and Wheat Improvement Center, we hold in trust two of the world's largest and most diverse collections of staples, namely wheat and maize. I'm going to call corn maize. I know most people here know it as corn. Coming from the UK, corn is wheat to me, so I'll just confuse myself. So you have to excuse the nomenclature. So within the maize collection at CIMMYT, we represent most of the areas of diversity, obviously the primary center of diversity and the center of origin, Mexico. So one of the questions that many breeders often throw to me is, well, why do we even bother looking at this gene bank diversity? Now I throw back this kind of image. This to me is what most of my breeding colleagues look like, okay? Prior to me being lynched as I attempt to leave here, let me explain why. Breeders over the years have done an exceedingly good job of working with plant genetic resources and pulling out those genes and alleles of large benefit. Okay, we've done a very, very good job. But now we're tending to fish from what is potentially an increasingly limited pool. And this really is that global picture of plant genetic resources. Okay, there's many more genes and alleles potentially there that we can start to sample. Now, why don't most breeders use germplasm bank materials, use accessions? Well, as my friend and breeding colleague Terry Molnar will often point out, there are many elephants in the room. One of the biggest issues is linkage drag. Okay, bringing forwards with any favorable genetic variation, deleterious variation and variation that you simply don't want. These are maize fields, maize plants that we're trying to use in some of the breeding work. Here you can see, okay, this is an ear height, yeah, and these aren't pygmies, these are normal sized people, okay? It's very, very difficult to work with this material. No breeder wants to touch something like that. Some less complex issues often are things like grain color. These are some accessions that we've identified from the gene bank that have favorable disease tolerance, but they're colored. The disease occurs in Kenya. Kenyans do not want to eat colored maize. They like white maize. So even though it's not an intractable problem to bridge that gap, it still involves a number of generations in addition to general use to try and clean these things up to a background that breeders are interested in. Working in maize, one of the other challenges is extreme diversity within the accessions that we're dealing with. This is a plot of flowering time, okay, from Martha Wilcox and Juan Burgueño. And this is within test cross flowering time. So we've taken an accession, we've developed a test cross, and this is the variation within plots of flowering time. 75% of the entries here have a variation of more than 10 days within plot. Okay, so even trying to make crosses with this germplasm is somewhat challenging. The other big challenge, and one I find the most intractable, is lack of relevant information. Okay, I think of a gene bank like this. Okay, most gene banks look like this to me. There's a lot of potentially valuable material in there, but like a supermarket with unlabeled cans, I haven't got a clue which of the 28,500 accessions in the gene bank is relevant for my use. There are many cases where gene bank accessions are reordered time and time again, and they might be the one or two things that someone's written a whiz-o publication on, and people go back and sample that same genetic variation. But by and large, there's very little information to help me make a selection. So one thing that we're trying to look at doing is trying to enrich this information, okay, so that we can go in and make an informed choice of which materials might be most relevant to start looking at. Now, coming back to the needle in the haystack, okay, I'm not necessarily interested in the haystack, I want the needles, okay? Slightly different paradigm and way of looking at things. We know from some of the initial work we've done looking at maize variation, within the elite maize material that we have at CIMMYT, material we call the CIMMYT maize lines, or CMLs, only around 6% of the allelic variation present in the gene bank is there. The rest of the material, the rest of that variation is rather like an iceberg. We're not seeing it, we're not sampling it. Now, much of that is probably useless. Yeah, not all diversity is good, and I'm never going to stand here and tell you that. But there are things here that are potentially relevant or potentially useful that we're not sampling. So how do we better do that? The other thing to consider really are the big challenges, okay? And these are challenges that everybody here is contributing to in one way or another. We know that the global demand for food is increasing, okay? With changing diets, changing population sizes, that pressure is becoming increasingly acute. The genetic gains in the developing world are at a they can spit test at about 1% a year. Okay, they're not particularly high. The challenge that climate change throws on top of that is increases in temperature, okay, changes in drought preference, changes in diseases. We see about a 5% yield loss per degree of warming. Now, as climate change continues, that means that this genetic gain that we try to make is constantly being eroded. It's like my kids when they suddenly get in the airport and start trying to walk forward on the moving walkways running in the opposite direction. Okay, we have to try and move that bit faster to keep up with the changes. From the CIMMYT perspective, okay, most of the CGE system, 45% of the global area sown to the major staples is found in low and low middle income countries. And these tend to be regions of the world where the private sector doesn't have a particularly strong focus for many reasons, understandable reasons. But these countries also co-locate with by far the majority of the world's poor. But as I mentioned with private sector investment, they only receive 5% of that funding. So the public sector has a strong influence in these localities. So with this issue in mind, CIMMYT leads a program called the Seeds of Discovery Initiative. It's part of a broader umbrella termed MassAgro, funded by the Mexican government predominantly. And it works on the interface between the genetic resources, the gene banks, and the breeding programs. There are obviously other initiatives within this, working on wheat and maize breeding, and then trying to package those new varieties, that newly formed genetic variation, with enhanced agronomic practices, better tillage practices, better fertilization rates for farmers. So the genetic resources aren't the panacea, they're not the magic bullet that solve everything, they're part of this broader continuum. Within Seeds of Discovery, we have 4 main pillars of work. We have work looking at the genotypic characterization of the gene banks, trying to provide a common framework to all of these accessions. We cannot possibly phenotype everything in there. Many of these accessions have bits of missing data. We might know what country they come from, but not exactly where. So we use genotypic data as a common backbone to help layer these other bits of information on top. On top of that, we have phenotyping activities. Okay, as I said, we're not phenotyping everything, we're phenotyping select panels, and then marker trait associations linked with those efforts. Pre-breeding or repackaging some of the alleles, and then some work on capacity development and obviously data management. And I'm going to talk to you about snippets of these first three today. So with the work in the genotyping, we try and view this almost as a satellite navigation system for genetic diversity. Now, many of us nowadays have sat-navs either in the car, or we have them on our mobile phones, or you have them on your tablets. And what these do is help bring together all those resources that you would have used 15 years ago. Okay, I used to drive around Mexico City with a map, trying to drive and read the map at the same time. Nowadays, we use the sat-nav, and hopefully it points us in the right direction. So it brings together the old map information. It tells me if there's a policeman further up the road with his speed gun waiting to try and capture me. It tells me if there's traffic information. So it brings together a large panel of information resources and helps me or another user make a decision of how do I want to get from A to B? What particular path do I need to take? So from the diversity side, we can view this in a similar manner. And we've tried to form and put together a system that we call the molecular atlas. So as I said, we use the genotypic data as a base framework for this, and layer on top of that other information sources that could help a gene bank user identify which particular accessions in the bank may be more relevant for their use cases. What we've done to date is genotypically characterize everything that we have in the maize gene bank. Okay, everything has been genotyped in there. And we've used a platform called DartSeq. Okay, it's a sequence-based extension of the old Dart Array platform. And this generates both single nucleotide polymorphisms and what we call silico darts, or presence-absence variation markers. Now, obviously, with maize, we can't just look at a single individual from each accession. We know there's a broad range of phenotypic diversity. There's a very broad range of genotypic diversity within an accession, very heterogeneous samples. So we worked with the data providers to develop a modified protocol of DART so that we can genotype a composite sample and look at allele frequencies. So we've genotyped in total around 35,000 entries, covering land races, pools and breeding populations that are present as accessions in the gene bank, wild relatives, so trypticums, teosintase, the CIMMYT maize lines, those elite varieties or elite lines that are present in the bank, and then a variety of donors, disease tolerance donors, drought donors, and ex-PVP materials that aren't necessarily available directly from the gene bank. And we're using this data for a whole array of different analyses, whether it's basic diversity analyses, the development of breeder-oriented panels of germplasm, selection sweep analyses, etc. From the diversity work, here you have representation of multidimensional scaling. This is of around 15,000 accessions present in the bank. We see the usual distribution of diversity that we would hope to expect. Here you have a big blob of Mexican accessions. You have South American, Peruvian-based material up here, a broad range in between. When we look at adaptation, we tend to see highland falling out at the top of the plots, and we see the subtropical and lowland tropical falling further down. So that's, when I talk to my wheat colleagues about the wheat data, the head of the wheat gene bank turns around and says, well, we already knew that. Great, I'm happy that we see the same thing that you already knew. That means some of the analysis is at least robust. So what we're doing with this in terms of breeder panels is trying to bring together some of the data that we have from the passport material and GIS informed information from this, together with the genotypic data to narrow down from that huge amount of material to something that a breeder could have used in the field. So within the gene bank, we have around 19,000 entries that have good geolocation data, information from their collection sites. And we can use that information to go back into the climate data, into soil data, and bring out long-term climate averages. So we can identify materials that have evolved and have been selected in regions subject to pressures that we're interested in nowadays: high temperature, drought, low soil pH. This panel represents around 5,000 accessions that have come from environments where during flowering you have very high temperatures. Okay, interesting, great, but it's still too many for me to evaluate. If a breeder looks at this, this would cost around $1 million to evaluate in a fairly conservative manner. But what we can now do is use the genotypic data to narrow this down a little further. So we can form panels. Okay, I'm not going to use the word cores because my gene bank colleague will kill me, but we can pull down panels. Here we have a representation of 1,000 of these accessions where we maintain some of that overall distance and diversity, but this is going to cost me $200,000 to evaluate. And we can come down further and further into something that's more affordable. So people can sample that diversity. If you find something interesting, say up here, we can go back through the panels and find more accessions that are somewhat related to that material of interest. Now this comes alongside some of the phenotypic evaluations that we've been doing. This was an experiment that was set up right at the beginning of the project. Okay, it's a GWAS panel. And here we took around 4,500 land races from the collection. So these were materials that had been screened somewhat before and were considered to have higher agronomic value. Okay, they didn't all grow and lodge in the field. So these materials were taken and a single individual per population was crossed to a single cross hybrid to form a modified test cross, a modified F1. This F1 was used in a whole series of phenotypic trials, and the DNA from the male parents here was genotyped, and this was genotyped using the GBS from Cornell. That data was imputed. Okay, we ran some imputation. Now, I'm not particularly an imputation fan, but as we've heard earlier, we have issues with trying to get enough statistical power with low marker numbers. And maize is probably as bad as half of the other candidates mentioned in that we have very rapid LD decay, so we need that marker density. Now this was work that we conducted with a student, Alberto Romero, who graduated earlier in the year. And he was working in Ed Buckler's lab, and Ed likes acronyms, so he's termed this FOAM, F1 association mapping. Doesn't sound very pleasant, but that's what we've got. Now, the focus of the phenotypic evaluation was very much on field-based phenotyping. Okay, I know there's many different ways of evaluating a phenotype, but from the CIMMYT perspective, we like to see things in a canopy in the field. Okay, initially that's the most relevant breeding target for us. And we screened across a range of different environments covering abiotic stresses, biotic stresses, issues of grain quality. But across all of these, we took some very basic metrics. So we had flowering data, information on plant and ear height, stem and root lodging. Now, the person conducting this phenotyping, Martha Wilcox, did a sterling job. We evaluated these materials across about 36 different environments. So that's an awful lot of phenotyping that was involved, generating just over 1.5 million different data points. So we initially looked at flowering time as a way of evaluating this approach as a proof of concept. We understand some of the pathways and genes involved in flowering time. Were we going to see them when we interpreted this data? And this was published recently in Nature Genetics a little earlier in the year. So, yeah, we've all seen the wonderful Manhattan plots. we see the usual candidates popping up. Okay, we've got ZCN8 there, VGT1. We see overlaps between the expression of some of these materials or the presence of some of these QTLs with other features. Because we have that GIS-related data, we can start to look at other factors. So we ran analyses looking at latitude, analyses looking at altitude, and we see a lot of overlaps. Now, one thing that did pop up, which was new to us, was an inversion on chromosome 4, which, I mean, Jeff and his lab have looked at this and documented this previously. So here you see this big inversion. This is an inversion that comes in from Highland Hyacinthe. And here we see it in flowering time. This is days to female flowering. Here you see the same thing popping up in response to altitude. Okay, so we get these big locations and co-location of a lot of these interesting QTLs. Now for inversion 4, as I said, this was new to us. Okay, this is an effect on flowering time that had never been documented, and it's the single largest effect on flowering time documented to date. So, okay, to most breeders, you can say we found QTLs for flowering time. They'll say, so what? I can measure that in the field. Okay, this isn't rocket science to me. But what this does demonstrate is proof of concept that the GWAS approach at least works for this complex trait. And in addition, we can find variation or find QTLs novel to those already described in the literature. So, as I said, we've not just looked at flowering time. Here is a pic for a Manhattan pond looking at grain protein content. As you can see here, we've got many of these skyscrapers that we can start to attack. Here you can see this inversion on chromosome 4 popping up again. This inversion seems to pop up everywhere for every single trait we can possibly look at. In addition to using direct phenotypic data, we've been exploring how far we can use some of the environmental data, okay, this GIS-derived data. And we've been using what we term environmental GWAS, so using this GIS-based data as a phenotypic proxy in the analysis, and we've been using this information in more standard selection sweep analysis. So this is something that Alberto popped up just before he was writing up his thesis. Here you see a Manhattan pot from chromosome 4, and we were looking here at soil pH, okay? Here you see an absolutely beautiful peak right on top of an aldehyde dehydrogenase. Okay, something we expect to be involved in acid soil tolerance. This is another plot. Here you've got chromosome 9. Here the factor put in was precipitation during the 6-month growing period. Okay, 6 months of that growing period where maize is from planting right the way through to harvest. Here you see a transcription factor for a heat shock protein. Okay, this is something that is of interest within industry in terms of gene editing as a target. So we can start to think about bringing these different data resources together in ways that we've never really explored before, and making some informed choices of which regions and which accessions may be of value. Now, all of this is very good, but if I still tell a breeder, okay, here is an accession that might have a fantastic allele in it, but the rest of it really, to be honest, looks quite crappy, they're still not going to want to touch it. Okay, we've got to try and repackage these materials, and most of the breeders that we work with, they want this interesting allelic variation in backgrounds that look more or less like the inbreds that they're working with. So what we need to do is develop inbred or semi-inbred genetic material, and not just semi-inbred, but material in backgrounds that resembles those elites that the breeders are actually targeting at the moment. And in addition to good agronomic performance or good performance under the trait, yeah, these things need to look reasonable in the field. They don't need to lodge. So pre-breeding, what is pre-breeding? Okay, when I mention pre-breeding to some breeders, they say, well, it's just breeding. Yeah, it is just breeding. It's just a bit earlier up the food chain. So I view an accession, a gene bank accession like this, okay? It looks like a cat. Okay, it has fur, it might purr, but I ain't going to take this home to my mother, okay? It's going to have issues. Pre-breeding takes these very wild and relatively undomesticated materials and generates something like this, okay? It looks like a kitty, it acts like a kitty, but it still looks a little bit rough around the edges. But here, this is something I could take home to my mum and she wouldn't be that offended with. The next process is handing over this kind of germplasm to a breeder, okay, who is then going to take that and they're going to develop the wonderful new varieties, okay, and add all the blingy things that you want in a modern variety in farmers' fields. So really we're interested in this transition zone. Now within the project we're working on pre-breeding for a number of different traits. Drought tolerance at flowering, okay, is a very big issue that we're trying to look at. We work at heat tolerance, at flowering and during grain fill. We've got some work that's relatively new looking at resistance to maize lethal necrosis. It's a viral complex that's really popping up and causing a lot of issues in Eastern Africa at the moment. We work on resistance to tar spot disease complex. Okay, this is a disease prevalent in Mexico, Guatemala, and it's moving north towards the US, and there's some cases of it in the US. It's a disease, fungal disease involving 2 to 3 different pathogens. And then we have a small project working with Kellogg's on maize with high anthocyanin content, okay, so for some high value end uses. Now for all of these materials, we've taken interesting entries out from the GWAS panel. We've done per se testing on accessions that we've identified from target environments. We use a lot of back cross breeding. So if you want to know more about this, please contact my colleague Terry Molnar. He'll be exceedingly happy to talk to you about it. And we also use genomic selection where possible. So just to give you a quick snapshot, these are some of the early entries that we've been evaluating from the back cross breeding program. So here is one of the best drought tolerant donors that we have going. Here, very reasonable yields under drought, still decent yields under well-watered conditions. These materials here are the best entries that we have. So these represent different accession backgrounds, okay, within the same elite, all showing pretty decent yields, okay, not exactly as high as the best drought tolerance donor, but this thing has been developed over about 15, 20 years, okay? It's gone through quite a lot of intense selection. These materials all yield higher under stress than some of the commercial hybrids that have been released through the Massagro maize breeding program. Okay, so we're starting to make some pre-breeding material that it has reasonable agronomic performance and is bringing forward variation for traits of interest like drought tolerance. Now the challenge is to identify what is underlying these traits. Okay, we know that these accessions are really very different to some of the donors that we have, but are we dealing with the same allelic variation? That we need to answer. So when we've been looking at different options, okay, how do we better target this breeding? How do we better move it forward in a faster manner? We've been looking at genomic selection. Now this is the results of some simulation work that we've done with John Hickey and Gregor Gorjank. And here we started to ask the question, what different breeding approaches, what different parameters really influence the response to selection within a landrace panel? Now we throw, my usual thing of throw so many questions at them. We ran the simulations, managed to break a computing facility that NASA uses, so we did very well. We're exceedingly popular there. But we've managed to narrow things down to a smaller set of parameters that we can use and help inform some of our pre-breeding work. So one of the initial ideas was to come back to test crosses that we'd made in the GWAS panel and start to use them as foundational material for genomic selection. And yes, the test cross approach here versus landrace or doubled haploids derived from landraces ultimately gave us higher genetic gain, But when you look at the simulations of what we'd essentially pulled back out, it's the elite parent, okay? Very few of those interesting novel QTLs were dragged forward. We're pulling out the things that the breeders have already fished out. Yeah, that's not relevant. So we end up following approaches which give us lower genetic gain, but help start to pull forward some of that novel allelic variation. And if you're interested, I mean, have a look at the reference, and when you have insomnia tonight, please go ahead and read this. Now, when we look at genomic selection, one thing that we'd mentioned earlier was marker density. And one thing popping out from our simulations was you want as much marker density as humanly possible when running genomic selection with broad-based land races. And this is the results of some analysis, just some of the cross-validation results from the GWAS panel. Now, the GWAS panel, in addition to genotyping with GBS, we genotype them with DartSeq, so we can actually start to ask some of these questions. And here you've got some data looking at plant height. Okay, we can look at days to anthesis, days to silking, yield. We see a pretty similar pattern. You can get, as we'd expect, across the different environments, depending on the heritability of traits, very different responses to selection, depending on the quality of the phenotypic data we've pulled in. But in terms of the marker density, we don't see a real difference. Okay, I'm really happy with this, as I can no longer use GBS, I can use DartSeq. Yeah, no problems there. So the question is, where do we go next? Well, with my increasing grey hair, I'm probably going to end up looking a little bit more like crazy Catwoman here. We've got some of this tame genetic variation we're bringing through, but what else can we do with that? One of the things we're starting to do now is see how we can combine the DartSeq data with the GIS data to conduct environmental GWAS. Okay, this is a bit more of a stretch because here marker density really matters, and we haven't got quite the marker density that we'd like to look at. But the other thing is then using genomic selection there, really to ask the question, can I pull out higher GEBVs from those gene bank accessions using GIS data as a phenotypic input? We're taking the DartSeq data as a way of asking this and looking initially at some of the GWAS phenotypes to look at genomic selection, moving that back into the bank, looking at things like flowering time and plant height. These are things that we've measured in the GWAS panel. We can use that as a training population, and coming back into the gene bank, whenever we regenerate these materials, we capture these kind of metrics. So I can actually blind test that population and validate to some extent whether we are getting an ordering of the genomic breeding values. So with that, thank you very much for your interest. I'd like to recognize the donors, okay, the Mexican government through Masagro, the CGIIR research program on maize, and then some funding and collaboration on informatics from the BBSRC in the UK. And thank you very much.
Any question? Thank you for the talk. That was very interesting. Is this on? Okay. Yeah. So your approach has been to kind of take phenotypic diversity, identify the regions that are affecting that, and then move that into your breeding program. Have you tried looking at it like the opposite way and said, these are the haplotypes that exist in our breeding program. These are regions where we have very low haplotypic diversity. Where can we go find diversity to move into those regions?
We're doing that in wheat, in maize. I mean, unlike many of the companies, we don't tend to have regions that have low genotypic diversity at the moment. It's very broad still within the CG breeding programs. In wheat, that's an area of particular interest, especially those areas where we tend to get very little recombination.
The same for soybeans too. We have a lot of forward breeding and fixation in the genome, so it's species dependent too. Thanks.
I assume that you're working with the inbred lines here. How many thousands of inbred lines that CIMMYT has developed over a period of time?
Oh, good God. In wheat, that's easier to answer because a lot of that material goes back into the gene bank. So there will be tens to hundreds of thousands, yeah, over 70,000 inbreds for wheat. For maize, that number is much lower. So the best material, the absolute best, gets forward to be a CIMMYT maize line. So there's 570. Yeah, we're quite restrictive with that. For donor lines, there's probably another 200 to 500 donor lines available, and then breeding lines within that. There's very many more.
Any other question?
Coffee.
Thank you, Sarah.